Discussed about lysosome. We all know that lysosome is a cytoplasmic organelle that is found in every animal cell and few in plant cell. So we can call lysosome also the suicide bag of the cell. So Christian D. Dave in 1995 named the lysosome because they contain digestive enzyme capable of tissue dissolution. Next, occurrence of the lysosome. Lysosomes are present in all animal cells but a few number in plant cell. These are most abundant in macrophages and in the cell of those organs which are associated with enzymatic action like liver, pancreas, thyroid, spleen, intestine and kidney. Next, structure of the lysosome. In different cell and even one cell, the lysosomes exhibit polymorphism. It exists in two basis morphology, forms as primary lysosomes and secondary lysosomes. That is primary lysosome and secondary lysosome. Primary lysosome. These are the storage granules formed either directly from the endoplasmic reticulum or from the Golgi apparatus. By the process of boarding, they are membrane covered package of at least six different clauses of enzymes. Then secondary lysosomes or phagosomes. These are the membrane bound vacuoles formed by the endocytosis of extracellular substances. Desired to be partially or wholly digested, they fuse with the primary lysosomes so that their enzymatic content can act on the engulfed particles. The newly formed vacuoles is called the secondary lysosomes, and intracellular digestion takes place within these vacuoles. Then from the secondary lysosomes, the residual body starts. So we'll discuss about residual bodies. These are the cytoplasmic vesicles which contain undigested or undigestible substances. Residual bodies are formed as the enzymes of lysosomes can't digest certain compounds, and these residues must be eliminated from the cell. In some cell type, they migrate to the cell membrane, fuse with it, and release their content into the extracellular space. Then autophagic vacuole. In certain conditions, some part of the cell, including some part of organelle as mitochondria, endoplasmic reticulum, are enclosed in the process of digestion. By some membrane bound vacuoles, they are the sites for breakdown of the parts without causing any damage to the intact cell. In liver cells, during starvation, autophagic vacuoles are found containing remnant of mitochondria. Next, some enzymes of lysosomes. That lysosomes contain tissue dissolving or hydrolytic enzymes that digest biological substances into smaller molecules. That is first nuclease. What is nuclease? These act upon nucleic acid and hydrolyze them into nitrogenous bases, phosphate and sugar. Then phosphatase. Phosphatase are acid phosphates and phosphoprotein phosphates that hydrolyze phosphate compound. Then protease. Protease or proteolytic enzymes. These enzymes hydrolyze protein into amino acids. These are cathepsin and collagenase enzyme. Next, glycosidase. These are beta galactosidase, beta glucuronidase and alpha monosidase. These act upon polysaccharide and alpha glucosidase. Next, sulfatase. These hydrolyzes sulfate ester into fragments. Next, lipases. These enzymes include acyl lipase and phospholipase. These break down lipids. Next, function of lysosome. The first function is The first function is digestion of external particle or heterotrophy. So, the intracellular digestion of exogenous material is called heterotrophy. Large molecules of food are ingested by lysosomes. The cell engulfs particles by forming an invagination which become pinched off from the cell membrane in the form of food vacuole. This process is known as phagocytosis or endocytosis. Next, second function is digestion of intracellular substance or autophagy. The lysosomes can digest parts of their own cells, and the process is known as autophagy. Autophagic vacuoles are responsible for several types of cellular activities such as cell turnover, cellular remodeling, etc. Next, cellular digestion. The lysosome can digest the entire cells in which they are present. This occurs in pathological condition or when the cell die. Next, sperm penetration. During fertilization, the acrosome of sperm head produces some lytic or lysosomal enzymes which dissolves the tissue around the ovum and help in the penetration of sperm in the ovum. Next, osteogenesis. During conversion of cartilage into bone, the special osteoblast cell produce lytic substances which erode the matrix of the cartilage and help in the formation of bone. Next, chromosome breaks. Lysosome contain in the enzyme deoxyribonuclease, that is DNA. These enzymes causes chromosomal breaks and their rearrangement.